Hopefully by now you have a pretty good idea of what happens during transcription, the splitting of the gene on the DNA and then the making of the mRNA in the middle of the DNA. Now we're going to take a look at the second part of protein synthesis, which is translation. There's a lot more to translation than there is transcription. The best way to start this, in my opinion, is to show you an animation of it and kind of label some things on the animation. But then we're going to go through this in your notes and we'll review all these terms again. I will try to put the link to this animation on Classroom, but just be advised, you have to open it in Internet Explorer. It will not work on Chrome. Again, just like with cell division, a cell is not going to replicate its DNA and divide unless it gets a signal to do so. Same with making a protein. A cell is going to make a protein when it gets a signal that it needs to do a certain job for the cell. So it's getting a signal that it needs to make a protein. So just the area of the DNA, the gene, that codes for the protein is going to get unzipped. This is showing you that in the middle of the DNA is where we make the RNA. And the RNA is made from RNA nucleotides. There's no T in RNA, just use. And then after the mRNA is made, you can see it leaving from the middle of DNA and it's coming out of the nucleus. You can tell that because one of these little holes is called a nuclear pore. So it's coming out into the cytoplasm. Now we're ready for translation. We have the mRNA, it's labeled for you in red, that's out in the cytoplasm, and a ribosome has attached onto it. The ribosome is labeled. Now every 3 bases on the mRNA is important. Every 3 bases is called a codon. And a codon is going to code for an amino acid. Remember we said proteins are made of amino acids. We have to string together amino acids to make a protein. This is how we're going to actually build the protein now out of amino acids. If you would, you need to turn to the next page in your notes where it starts translation. And you're going to see in the bottom of that page, there's a codon chart. So take a second to try to find this chart because you're going to look up some things. So I'm going to move this down. And this is what I mean by the codon chart, the mRNA codon chart. So you're going to look up some things as I'm going through this animation. GGC is the first codon on the mRNA. I want you to go to that chart and I want you to find GGC. GGC is found right here and it's coding for this amino acid. That's an amino acid. Actually, all these are amino acids. This one is abbreviation for glycine. You don't have to know what gly means. You just have to know it's an abbreviation of an amino acid. Now, this next part shows you, here's the amino acids that are in the cytoplasm of the cell. You eat protein and you break your proteins down into amino acids, and then these amino acids are shipped to your cells so you can use them to rebuild your own protein. So here's the amino acids. They came from your food that you ate. This one right here is glycine. That's what it's trying to tell you, is GGC codes for this amino acid. Now these amino acids just don't float together and form a chain. There's going to be another molecule that you need to know that's going to assist in translation. And it's these guys. These are the tRNA molecules called transfer RNAs. I think it's easy to remember them. They kind of look like a T here. So think of that kind of cross or that T. They are going to go pick up the right amino acids. So this first tRNA went and picked up glycine and brought it to this ribosome that is hooked onto the mRNA. So again, let me just emphasize that this is a tRNA molecule. And this is the first amino acid. Specifically, it is glycine, G-L-Y. The next codon is AAC. Or if I said, what is the next codon in the sequence? You should be able to tell me AAC is the next codon on the mRNA. AAC, if you go and you look it up on that chart, AAC codes for a specific amino acid. Another tRNA is going to bring that next amino acid. AAC codes for, I believe this one is ASN. I think that That stands for like asparagine. You don't have to, or asparagine, you don't have to have that memorized, but this is the second amino acid in this protein that we're trying to build. If you notice, you'll see that one amino acid gets transferred to the other, and that first tRNA just left. There is now a bond between these two amino acids, and you know from the biochemistry unit that amino acids are held together by peptide bonds. And as we continue to make this animation go, you see the next tRNA comes, and it's going to have the third amino acid, and so on. And we're just going to continue to bring in tRNAs, bringing those amino acids, and we're making a protein. So this is one protein that is made right now of 6 amino acids. Now, I'm jumping back in this animation to talk again about these these tRNA molecules. And you can see labeled here, tRNAs have 3 bases on them. And those 3 bases are called the anticodon. You're going to see that the anticodons are going to match up to the codons on the mRNA. So again, you already witnessed this, but I just wanted you to understand that here is a codon, and then this part right here is called the anticodon of the tRNA molecule. And notice the bases. G goes with C. G goes with C. C goes with G. So let's envision this next tRNA coming in with their amino acid. And let's think about what would be the anticodon that would hook up to the next codon. The next codon is AAC. So this tRNA, and the only tRNA that can hook up to AAC is one that says U, U, G, because A can hook up to U, A can hook up to U, and C can hook up to G. Now let's go ahead and move to this page in our notes, and then ultimately we'll go back and do some of the practices on the previous page. Again, we are on step 2 of making a protein, and this is called translation. And in translation, we're trying to figure out the right amino acids to put in the right order so that we can build a protein. We say that translation happens at a ribosome in the cytoplasm, which we already mentioned. And another way that you can say this is the mRNA is being read during translation, or you can say the mRNA is being translated, not transcribed. If you said that RNA is being transcribed, we're back in the nucleus and we have the DNA split open and we're building mRNA. Once the mRNA is at the ribosome, though, we say that it's being translated by the ribosome and a protein is being built by the ribosome. Remember, ribosomes are going to make proteins by putting together amino acids. We mentioned before that whenever you make a protein incorrectly in your body, then you are born with some kind of genetic condition. So it's critical that our DNA codes for the correct sequence of amino acids. If you put in one wrong amino acid, then your proteins won't fold correctly. They'll misfold. They can't do their job. You end up with a non-functioning protein. And if you remember from the first pages of our notes, that is what is causing all these genetic diseases that these people are born with, is they inherited a gene that coded for the wrong protein. So a person with cystic fibrosis, they cannot make this transport protein in their lung cells, so therefore they have this genetic disease. A person who is albino, they do not build melanin correctly, so therefore they suffer from albinism. So it's critical that we put in the right amino acids and that our DNA codes for the right amino acids so the protein will fold and it'll function and do a job for the cell. Okay, moving back here to where we're at. Now let's kind of break this down step by step. So here's the events of translation. We already said the mRNA, which you see here in purple, it has arrived at a ribosome. So you should recognize that this blob right here, that's the ribosome. And you can tell according to our drawing here, we already have a tRNA and it is hooked up to this codon. Now what's kind of interesting is you notice this tRNA, it didn't start with AUU. it hooked onto AUG. So one thing that you need to know is that whenever translation starts, the first tRNA always goes onto the codon AUG. AUG is called the start codon, and it's the only start codon. If you look at it on that codon chart at the bottom of the page here, you're going to see it says this is the start codon. So when the ribosome clamps down to the mRNA, it's going to go and find the start codon. The start codon is AUG. And remember, every 3 bases on an mRNA is called a codon. It is the job of the ribosome to match up these tRNAs that are coming with amino acids to the mRNA. And that's going to bring these amino acids close enough together that they're going to form that peptide bond there. So the ribosome matches a tRNA up to the mRNA. Remember that tRNA is carrying what we need to build proteins, which is the amino acid that's going next into that protein. The amino acid that it is bringing is determined by that mRNA codon. So for example, I want you to go down here to the bottom and look up GCC. This is the next codon. When you look up GCC, you're going to see ALA next to it, ALA. That's alanine. And you can see that this next tRNA that's coming, that's what it's bringing. It's bringing the next amino acid, alanine. And this tRNA is going to land and match up right here, just like the first one did. Now, let's talk anticodons. Anticodons, or the anticodon, would be the 3 bases on the tRNA that are complementary to the 3 bases on the mRNA. So if the mRNA codon is GCC, you have to be able to tell me that the anticodon on this next tRNA would be CGG. Let's label this is the anticodon on the tRNA molecule. To emphasize, the next one is, the next codon down here is UGG. The first 2 tRNAs have already landed. The third one's coming. If you look up UGG, you can see that it would be coding for this amino acid, tryptophan. And the anticodon that you would find here on this tRNA molecule would be ACC. That's the anticodon. So to fill in that part of our notes, it says if the codon reads GCC, then the tRNA is going to bring the amino acid ALA. If the codon, which was the next one, the third one, was UGG, then the next tRNA that's going to land is going to be carrying tryptophan. And the tRNA can hook onto the mRNA because the tRNA has the anticodon, the 3 bases that are complementary to the mRNA codon. Remember, we're talking about tRNA, and RNA, we already said, does not have thymine in it. It's going to have U's in it instead of T's. If I ask you a question like this, if the mRNA codon is AUG, the anticodon on the tRNA is, you got to be careful. Do not look up an amino acid and give me the name of amino acid here. I want the anticodon. So if AUG is a codon, then UAC would be the answer to this question. It would be the anticodon. Again, whenever these amino acids get close enough together, then you get that bond that forms between them. And that is a peptide bond that you need to remember. And as we come down here and we see that the ribosome has moved down the mRNA, more and more tRNAs are coming. That very first one that was carrying methionine, here it is. It's leaving. And it's going to bind with another methionine, and it's then going to aid in translation and the building of a different protein in the cytoplasm. So these tRNAs aren't destroyed after they're used. They just simply will go and bind to another amino acid that they can bind to. Now, before I end this lecture, I want to make a big deal about something, because I'd hate for you to do this worksheet and you write down about 25 amino acids and every single one of them is wrong. I want you to go to this codon chart, and I want you to circle mRNA about 12 times. The reason why we're doing that is because on this assignment that you're going to do in a second, what often happens is that students will not look at the mRNA codons to look up these amino acids in the circle. It is the codon that tells you what to put here. And unfortunately, students start looking at the tRNA anticodons. They'll look up these 3 bases, but if you look up those and put that here, then this is going to be wrong. That chart in your notes is an mRNA codon chart. We could have made a tRNA anticodon chart, but we didn't. We just made an mRNA codon chart. So you have to make sure that you keep your eyes on the mRNA and not the anticodons when you're figuring out the amino acids that we're stringing together here to make this protein.